On behalf of the Indian Academy of Sciences, I'm pleased to welcome you to the public lecture today. The Indian Academy of Sciences was founded in 1934 by Sir C.V. Raman with a goal to uphold the progress and the cause of science in the country with a strong connect with the society. In 2009, the Academy completed its 75 years, and in its Platinum Jubilee, the Jubile Chair Professorship was instituted. And this year, we are very happy to have Professor Rose to visit us as a Jubilee Professor of the Academy. It's very highly appropriate because it's the centenary year of G.N. Ramachandran's, and also the topic that you will talk about or you have been working on is of great interest to a wide range of fellowship in the Academy, from physicists to biologists. It's a pleasure to hand over now to Professor Balaram to chair this session and introduce the speaker. For you.
Just say one or two words about gender. So anyway, so firstly, again, we are very grateful to George for coming here. And in the past 1 week, he's given, I think, 6 lectures all told, and he's going to give several more. He's 82, and he's not a politician. So these are all good things. And then, thanks to George's presence, we have had two very rare sightings. So the first one was we went to Kabini and we saw a tiger. And now the second one is here on our own campus. We have Professor Balram, who is... But anyway, so let me welcome Professor Balram to come here. And it's a pleasure to have him back on campus. And of course, I'm sure you all know what a distinguished gentleman he is. He did a lot of work at MBU for many, many years on peptide conformation, on unnatural amino acids, and a huge variety of other things, including the Ramachandran plot. So it's I'm particularly glad that he agreed to come here and chair this session.
Good afternoon. Let me welcome Professor Rose to the Indian Institute of Science, and also maybe welcome myself back once more. When I was here for many years as the director, I used to say that directors should rarely be seen and almost never heard. And ex-directors, of course, are best neither seen nor heard at all times. So it's a pleasure to come back here again, and I'm really happy that the Academy has given me the opportunity to chair a session in which Professor George Rose will be delivering an Academy lecture. And it's a real privilege for us to have him here as the Academy Platinum Jubilee Chair. A word about our speaker this afternoon. Professor Rose has had a long career in the field of protein folding. And I first began to see his papers sometime in the early 1980s, when a great deal of interest was focused on chain reversals in polypeptides, and people were talking a great deal about the Venkatachalam beta turns and so on. Growing up as I did in the shadow of the Ramachandran plot, it was, of course, over time, a real pleasure to read some of Professor Rose's papers. I still remember one phrase that Professor Rose used in a more recent paper, I think it was a 21st century paper, on the Ramachandran plot, where he called the Ramachandran plot a profound organizing principle of structural biochemistry. And I think there was no better way of describing Ramachandran's work. Professor Rose has contributed extensively to protein folding. His contributions have always been very incisive and very thoughtful. In the late 1980s and all the way till the late 1990s, he published some very influential papers on recognizing motifs in proteins and on signals which allow you to understand where a protein helix will begin to terminate and so on. These sorts of papers really set the tone, I think, for much that was to follow in our analysis of protein structures. But I must tell you one story about Professor Rose. In the literature, I don't know him well because I've met him many times, but in the literature, he did put out a challenge. It was called the Paracelsus Challenge. And the challenge was this, that you take a protein and you change all the residues. You can't change more than 50% of the residues. And you must change it into a completely different fold. It'd be like taking an all-helix protein and converting it into an all-beta protein without changing more than 50% of residues. And you know, if two sequences have 50% identity, we know that they're practically identical as far as their structure and function is concerned. This was a challenge. And of course, to back up his challenge, I believe he put up, and this is what I read in the literature, I believe he put up $1,000 and said that whoever solves this problem will get $1,000. And Lynn Reagan at Yale did solve the problem. Maybe not entirely to Professor Rose's satisfaction, but nevertheless enough to take away his $1,000. So he wrote a perspectives article in Nature Structural Biology in which he said that maybe the next time we put out such a challenge, the prize should only be a T-shirt. This was in the late 1990s. 25 years on, I think the entire field of protein design and the entire field of protein structure prediction has undergone a major revolution. It's not a revolution that those of us who are old would have anticipated in the 1990s. Today, with the use of artificial intelligence, what Professor Rose in his abstract terms, machine learning, aided by neural networks and so on, you can do all kinds of amazing things. David Baker's work and the more recent work on the AlphaFold have completely transformed the way everybody looks at protein structures and the area of protein folding. Sometimes, of course, I think they've got the term protein folding wrong because the protein folding problem was something different. Nevertheless, this has had all the people beyond, I think, a certain age worried. And I think it's those worries that Professor Rose will amplify on and encapsulate in his lecture today. He's going to talk to us about science, thinking about, and then in parentheses, thinking about science. Professor Rose.
Thank you. What a kind introduction. Thank you. It's a great privilege to be here. And I too grew up in the shadow of the Ramachandra plot. So today I'm going to address something that is not my own research. Today I'd like to try to put things in perspective given the recent advent of artificial intelligence and in particular the success of artificial intelligence in the recognition of protein folds from amino acid sequences. And as Professor Balaram hinted at, in case you didn't get it, it seems clear that this is not exactly the protein folding problem, but it is certainly the protein fold problem. So more about that soon. So with that in mind, let's see if we can begin. So the perspective. We are now about 5 centuries from the beginning of the way that science has been done, reductionist science. And it has worked extremely well. And I'm going to talk about one particular piece of reductionist science today. I'm going to talk about Newton's laws, particularly because I think everybody in the audience knows Newton's laws. So let me try to use that as a paradigm to put things in perspective. And then I'm going to turn to large language models and in particular to AlphaFold, which is this artificial intelligence model that you give it the amino acid sequence, it gives you the protein structure, it's been enormously successful. And I'm going to do this from a Western perspective because all I can do is do it from a Western perspective. I recognize this may not be entirely an Indian perspective, but I think science is a universal language, so probably will be okay. So there are times in history in which a number of disparate influences, a number of different disparate directions, all come together and seem to make sense. And one of those times was the Enlightenment. I should say another one of those times was when the genetic code was elucidated. That was a great time to do science. But I'm going to turn to the Enlightenment because during the Enlightenment, that's a time when we really began to think differently. It's how we learned how to learn, in particular, how we learned how to learn science. And here are some famous names from the Enlightenment, and there are many of them. So I'm going to talk about Newton, but here is Boyle, the gas laws, Voltaire, who wrote literature, philosopher, Jean-Jacques Rousseau, the social contract, Ben Franklin in the U.S., who was instrumental in starting the U.S., economist, philosopher, musician, oxygen, astronomer, and so forth. All of these people doing different disciplines, and yet if you look a little bit, if you back off just a little bit, you recognize that they're sort of feeding from the same common views of things, and in turn, shaping our views of things. So how did we learn how to learn about the world? It really goes back, at least in Western culture, to the Greeks. And from the Greeks to modern science in the Enlightenment. And the question is, why did it take so long? And we skipped a slide here. The Greeks started believing in gods, multiple gods, and when bad things happened, it was because you had to appease the gods, that kind of thing. So science really started, at least the science as we know it, really started with the Greeks. And a book by Carlo Rovelli, a quantum gravity guy, on Anaximander, tells us a lot about Anaximander, who he characterized as the first real scientist. Somebody who thought that the way the world worked was not because of the will of the gods, but because of abstract, non-human, scientifically observable kinds of forces. And of course, everybody knows Pythagoras, the profound influence of Pythagoras. I mean, all the way from understanding how the Greeks could begin to figure out the size of the earth just by the Pythagorean theorem, all the way to, Pythagorean theorem is there in special relativity. So this has had a profound effect. And so forth. Euclid, who really taught us the way to start with axioms, and then we can, with this Aristotelian logic, begin to deduce things that are not at all obvious, and yet we can prove them in this sense in which we start with axioms, use logic, and we can arrive at a proof, something that is absolutely convincing. It is the basis of the way that we do mathematics. And Epicurus, who thought that it isn't the will of the gods, but it is really some indivisible things called atoms. I'm sure he didn't mean by atoms the same thing that we mean by atoms. I don't know what he meant. But he said that the way they moved was by love and strife. And you say, love and strife? Well, if you translate that to attraction and repulsion, now it begins to sound pretty modern. So, and so forth. And what the Greeks didn't know was they didn't do experiment. They believed, it's not that they overlooked it, They believed that you shouldn't do experiment, because when you do an experiment on some subject, some issue, you begin to perturb it, and then you don't see in it all its purity. You see a perturbed value of it. And if you want to know how it really works, if you want to really characterize it, then you don't perturb it. So for that reason, you don't do experiment. And so in a certain way, that makes sense. It's Platonic ideal forms. But on the other hand, you recognize some shortcomings, sometimes even amusing shortcomings. For example, Aristotle thought that men had more teeth than women. Now, it would have been really easy to ask Mrs. Aristotle to open her mouth and count her teeth, but he didn't believe in doing such things. The other thing that the Greeks didn't have was ability to quantify things, the same way that we tend to quantify things. So for us, experimental, especially for us who do theory kinds of things, experiment is the adjudicator of truth, but that was missing. And finally, what the Greeks really lacked was the kind of instrumentation that we all have at our disposal. You come to a place like NCBS or IIT and find that they've just spent $3 or $4 million buying a cryo-EM. The Greeks didn't do such things. I mean, amongst other things, there weren't cryo-EMs. So it did take us so long to get from Greek science to current science. And there are a couple of really nice books that I would recommend. One is by an Anaximander, written by Carlo Rovelli, the quantum gravity scientist. And he is an excellent writer, written many books, and he's written about Anaximander, who he characterizes as the first real scientist. And then Stephen Greenblatt, who's a professor of humanities at Harvard, and he wrote The Swerve. And The Swerve is about the Pope's secretary who was wandering around to various monasteries at the time. And in one of them, his name is Poggio Bracciolini, and in one of these monasteries, he discovered a book, the only extant copy of Lucretius. And that's how we know Lucretius, and that's how we know so much about how science began to break away in Greek times from this view that everything was controlled by the gods. There was so much of this literature that was lost in the eruption of Mount Vesuvius in 79 AD. Interestingly, it is science again that's helping us retrieve some of those lost manuscripts. They were basically burned to a crisp in Herculaneum, but nevertheless, that burnt to a crisp residue is still there. And now we're being able to, not we, people who know how to do it, are able to read those manuscripts without ever unrolling them. And you can't unroll them because they crumble away, but you can read them. And so you can read them with optical techniques that let you probe them. You get little hints, and then people who knew about the times and are fluent in that stage of Greece can begin to put them together. So who cares about any of this? And of course, we care. Obviously we care or we wouldn't be here. But we also care, everybody cares, because anti-science rhetoric has become so increasingly dangerous and it's threatening. So in the United States, we have a large fraction of the populace who believe that global climate change is a hoax. That is an awfully dangerous view if those people get into government, and they have. And so that's just one extreme example, but there are many examples of this. So anti-science rhetoric is a truly dangerous thing. So that's one reason why we care. What is science? Why should we be afraid of it? It's open-ended, it's rebellious, it's subversive, but it's not deconstructionist. It's not something that is trying to confuse us. In fact, just the opposite. So we value science for the same reasons that we value education. It allows us to see the world with greater clarity, and it results in a more meaningful life. And of course, there are a lot of practical reasons too. So, now, how do we acquire knowledge in science? And that's what this is going to be about. So you can be an honors chemistry student at Johns Hopkins, which is a pretty good university, and you can have an A+ average, and it doesn't mean you know how science is acquired. What it really means is that if you take the problems that are assigned, where the answers are in the back of the book, you know how to get those answers. So you know how to use science, but you don't necessarily know how it's acquired. Oh, well, maybe you do. So what is science? Here's the grade school definition of science that's taught to people like me when they're little kids. A scientist is someone who forms a hypothesis which is tested, and if it passes the test, then it becomes a law. And I mean, that's not wrong, but it's pretty glib. It's pretty superficial. So, and in a sense, it is wrong. So we have Newton's laws and we have Darwin's hypothesis. But it could just as well be Newton's hypothesis and Darwin's laws. So we begin to convince young people that there is something that is immutable about all this, providing it passes certain tests. What is it really? All of us are working in this area in one way or another. A realistic definition of a scientist is somebody who mucks around trying to explain how things work. And I don't know how to say it any better than that. But it is my experience, and I think it's everybody's experience. And here's the question. A theory in science can never be proven. So how can we trust a scientist? How can we trust science? I mean, a theory in mathematics can be proven by the very nature of mathematics and what we mean by a mathematical proof. But science, we can't prove it. So how can we trust science and scientists? So it helps to look into what the life of a working scientist is. The life of a working scientist, and this is certainly not your high school definition, is it starts with some kind of question. You're curious about something. Or you're trying to get a PhD and you have a smart advisor who tells you, here's a really interesting thing to work on. And you say, yeah, that really does sound interesting. I'll work on it. So it starts with a question. And then when trying to figure something out, and something complicated and deep, it often happens as you do it that you take little steps and you happen upon some results that seem to falsify the idea that you had. I will dignify it by calling it your hypothesis. And these results seem to falsify that. And somehow you know, and I don't know how you know, but somehow you know, don't take no for an answer. Because I think that if I really understand this finally, it will all be explained. And no will not be no anymore. So you keep plugging along. And finally you'll reach a stage, if you're lucky, where you really do understand what you think is the answer to your question, or at least a sufficient answer to your question. Maybe not every answer, maybe not the total answer, but it's sufficient to explain a large set of the things that you were asking. At that point, you say, okay, now I'm going to be my own skeptic, because if I'm an honest scientist, I will be a skeptic. And in particular, I know that it's really easy to fool myself because I'm so excited here, but I'm going to force myself to be a skeptic. And now, instead of not taking no for an answer, I'm not going to take yes for an answer. And I will try as hard as I can to show that what I just concluded is wrong. Psychiatrists might call this kind of pursuit obsessive compulsive disorder, but we call it living a privileged life. So science as we know it, I'm going to start by reminding you of something that I think everybody knows, and that is how science has been done, especially since Galileo. And it really works in a kind of set of phases. Um, the first thing that happens is we observe something of interest, and we observe it as carefully as we can and as quantitatively as we can. And as we observe, finally, if we're lucky, we begin to see patterns in it. And from the patterns, we develop theories or models. I don't think that we really ever know how the universe works. We don't know what reality is. I'm not sure that there is such a thing that is available to us called reality, but we have models of reality. Um, we have models, and some of them are very far reaching, and they work so extremely well. And so here are examples of models that go through these three steps. There's Tycho Brahe, who wants to understand how the heavens work, and using optical spectroscopy, namely his eye, he's mapping out where the planets are as a function of the time in the year. And he accumulates reams of data. And all he's doing is carefully noting where the planets are. And then somebody who works for him, Kepler, Johannes Kepler, recognizes that you can describe all that data with three equations, three mathematical equations. So those are Kepler's laws. And why do those laws work? Kepler doesn't try to explain why they work. Kepler only shows that his three laws describe these reams and reams of data. So that's pattern recognition. And then along comes Newton, who knew Kepler's laws and turns it into first principles, Newtonian gravitation. And there are lots of other examples, and a few examples that are truly transformative, like Michelson-Morley, leading to empirical Lorentz transformations, and out pops at the end general relativity. And the observed spectral lines seem to have empirical regularities in these lines, ... and you get quantum mechanics. So here are examples, and all of these follow exactly these steps. And that's typically how science has gone for about 500 years. And so the one I'm going to talk about today, and only one, is Isaac Newton, who really set the stage for modern science. And I want you to recognize that models, or scientific models, are often counterintuitive. In fact, they're most often counterintuitive, but we validate them by doing experiments. And if the experiment validates it and the experiment is sufficient, then the model seems to be validated and we begin to accept it and use it. But they're mostly very counterintuitive, including Newtonian gravitation. And they are models. Newtonian gravitation is a model. For a few hundred years, it was taught as, no, that's how it really works, until Einstein in 1915, with a more appealing model, a superseding model, if you will. So what I'm going to talk about, I got from Richard, a book called The Cure for Physical Law, and Feynman, in turn, got it directly from Newton, from the Principia. And I will argue that really these are models. They're not things in itself in the sense that Immanuel Kant meant. So a big point is that seeing is deceiving. They are truly counterintuitive. The sun doesn't really go around the earth. It looks that way, but we're pretty convinced that it doesn't. Almost all science is counterintuitive. The one thing that's not counterintuitive, the one discipline that's not counterintuitive, is thermodynamics. My ice cube, if I put it out in this room, will melt. Strangely, that seems to be about the hardest thing to teach, perhaps because of entropy. I don't know, but that's the experience. So I'm going to talk about Newton and this three steps, Brahe to Kepler and on to Newton. So Newtonian physics are in science, and here is Newtonian gravitation, a very simple equation. And it says two bodies exert, and you all know this, two bodies exert a force upon each other, which varies inversely as the square of the distance between them, and it varies directly as the product of their masses, and there's some kind of constant, which we'll call G, for the gravitational constant. And there it is. One of the most profound equations. And Newtonian mechanics, because it emerged from Newton, had a much bigger impact than it had emerged piece by piece over a couple of centuries, as many other scientific disciplines did. And so it had a big impact because it emerged entire from one man in a short period of time. And also the other Newtonian equation, F equals ma, or A equals F divided by m. A body reacts to a force by accelerating. It's changing its velocity every increment in time to an extent inversely proportionate to its mass. It changes its velocity more if its mass is smaller. Smaller this, the bigger that. Okay, so that's all you need to know. And I'm sure everybody has done that. Everybody in this audience has done this at one time or another. So, bodies attracted to one another by an invisible force, regardless of the distance between them. That's what this says. No matter how far apart these bodies are, they're attracted to one another by a force. And the force is invisible. And now we've all accepted that as being a valid part of the model. We've wrapped our intuition around it. But if you were to walk up to some person on the street who hadn't had high school physics or something, and you said, I think I know that any two bodies in the universe, no matter how far apart they are attracted to one another by a force, but it's invisible, you can't see it. And they would look at you and I don't want anything to do with that crazy guy. But that's Newtonian gravitation. So how did Newton come by this idea? And by the way, it's not that it just seems like a really ridiculous idea, unless you have shown that it works. It seemed like a really strange idea to Newton too, not just to us. So, if universal gravitation is right, and planets go around the sun, you immediately ask questions. What kind of path around the sun? How fast do they go? There are a lot of theories. And Copernicus already knew that the sun doesn't go around the earth. And in fact, although it was lost for a long time, Greek Aristarchus also knew that in this era about 200 or 300 years before the Common Era. It tended to be lost, but he figured that out too. So here we go with the three steps. Observation, the beginning of modern science. Tycho Brahe, the Danish astronomer, recorded exactly where the planets appear in the sky to distinguish one theory from another. And pattern recognition. Kepler, German mathematician. He was an astronomer. He was an assistant to Brahe. And the planets go round the sun as an ellipse with the sun as a focus. And he's able to recognize that just by plotting the data and using his equations. So, if the planets go around the sun as ellipse, let's look at it. Here is an ellipse. There's a focus. What's an ellipse? If you take 2 points and attach a string or something to 2 points, and it's longer than the shortest distance between the points, and you take a pencil and you push on the string and you start to push it around, you get an ellipse. and it has a long axis and a short axis, and here's a focus and a second focus. And the idea is planets go around as an ellipse. Here's the sun, and the sun is at one of the foci. So, Kepler's three laws. The orbit of the planet around the sun is an ellipse. Equal areas are swept out in equal times, and you can prove that at this point. But Kepler didn't prove it, he just observed it. And the time a planet takes to go around the sun is related to the size of its orbit, and it varies as the square root of the cube of the size of the orbit, where the size is the diameter along the long axis. So those are the laws, and now what can you do with it? Newton knew the laws. So here's the question. Does the planet go faster when it's near the sun? So Newton knows the laws. Does it? Yeah, it does. Because equal areas are swept out in equal periods of time. And so if this is one period of time, if this is an area, they're all the same. And this area has to be swept out in the same length of time as that. And so the planet has to go faster in order to sweep out an equal area when it's close to that focus where the sun is. Now, I think I'm not telling you anything you don't know. But in high school, when we learn these kinds of things, we learn them in order to be able to answer the problems in the book. We don't learn them as a way of comprehending how the world works. And there's something much deeper than knowing the formulas. It's trying to understand what the formulas are representing and the concepts behind the formulas. That's what we tend not to do. So now the big question, Newton's question. What makes the planets go round the sun? And here's what Newton knew. Kepler was curve fitting. Newton wanted to know. So Newton knew Galileo's principle that if an object is acting on it and is going along at a certain velocity in a straight line, it will continue in the straight line forever. How did Galileo ever come up with that? I'm sure he never saw an object moving forever in a straight line. So this is an example of conceptual science. You start out with an idea. His idea is that if you take away all the other forces, like friction, and set a body in motion, it will just continue in motion. Nothing will stop it. Now, why is that true? Why does this law of inertia seem to be true? And the answer is, no one knows. It just is. And I tell you, I would have been so happy if the time that I had learned Newtonian mechanics, if somebody had said exactly that last sentence to me, because I spent so much time trying to find out why it is that the mass keeps moving along in a straight line forever by deriving it from F equals ma. So it doesn't work that way. Okay, so it moves in a straight line. But says Newton, what if it doesn't go in a straight line? Well, if it doesn't go in a straight line, there had to have been some force that perturbed it. And so a force could be measured by the product of two effects. How much does the velocity change in a small interval of time? Namely, how much does the acceleration change in a small period of time? And so when multiplied by the coefficient called the mass of the object, that altogether is the force. And a way of measuring the mass is by the force it needs to change its velocity. So if a planet is going around in a circle around the sun or in an ellipse, and no force is needed to make it go sideways towards the sun, tangentially. But it would just keep coasting along. But the planet doesn't do that. Instead, it keeps deflecting towards the sun instead of coasting along. And it's deflected towards the sun. Essentially, the planet falls towards the sun. Am I in your way? So, this is I'm going to demonstrate it. You know the planet falls towards the sun, but In fact, there's always some kind of force that keeps it from going around in the circle. So here is, I'm the sun, and it goes around in a circle. And if I were to let go of this, it would probably fly out and hit somebody in the audience. I promise not to do it. But the reason it doesn't is because I'm pulling on it. And the reason that the planet goes around the sun is because the sun is pulling on it. And so the sun has to be pulling on the planet and forcing the planet to move towards the sun. So to get this falling operation, there has to be a force. And the origin of the force, says Newton, is the sun. So Newton showed that the statement, equal areas are swept out in equal times, is a direct consequence of this simple idea that all changes in velocity are directed exactly toward the sun, not just in a circle, but in elliptical cases too. And so from telescopes, Jupiter's satellites go around Jupiter. Any solar system, the planets go around the center of the solar system. Our moon moves around the Earth in the same way. In general, everything is attracted to everything else. The Earth pulls on the moon, the sun pulls on the Earth. Of course, just as the sun pulls on the Earth pulls on the sun too. We know that, but it's more in the sun's direction. It's bigger. It's more massive. So we know the Earth pulls on things. We know the Earth pulls on things because I didn't hit my head on the ceiling. I just came back down. So Newton had a thought experiment, and thought experiments, Gedanken experiments, have played a key role in science. And the thought experiment, we can figure out how far the moon falls in 1 second because you know the size of the orbit. So it takes about a month to go around the Earth. If you figure out how far it goes in 1 second, you can figure out how far the circle of the moon's orbit has fallen below the straight line, the tangent, it would otherwise have if it didn't fall towards the sun. And that we can do that calculation, and it turns out to be a 20th of an inch. So now, here's the thought experiment. Suppose that instead of the moon being 240,000 miles away from the Earth, the moon was on the surface of the Earth. So, of course, it has to be a thought experiment, otherwise it would knock into things, but never mind that. Let's assume that the moon is moving around the surface of the Earth. So, the surface of the Earth is about 4,000 miles away from the Earth's center. The radius of the Earth at the equator is about 24,000 miles. So, where we're standing is about 4,000 miles from the Earth's center. The moon is about 240,000 miles away from the Earth's center, and so the moon is about 60 times further away from the Earth's center than we are. So if there were another moon, a pseudo-moon, moving around the surface of the Earth, and if the inverse square law is effect, then an object at the Earth's surface should fall in 1 second by what it does for the moon in its current orbit, times 60 squared. It falls off as 60 squared, as the radius squared. So, and the radius between our distance and the moon's distance is 60, so it should be 3,600. And if you do that little calculation, it means that at the surface of the Earth, the moon falls, the object falls at 16 feet per second. It was already known from Galileo's experiments, dropping balls from Pisa, that these balls fall at 16 feet per second. So, incredible. Now, there's another test, that the pull of gravity is exactly proportional to the mass. So, the force of gravity is proportional to the product of the masses, and we're just talking about changing the mass from one to another, and it's the sun. So it doesn't change its mass. If it's proportional to the mass, it doesn't. The motions induced by the force are inversely proportional to the mass. Here, they are inversely proportional to the mass, giving rise to the acceleration. And so, in other words, these two different things, if there's a vacuum, no friction, that kind of thing, then regardless of the mass, when directly proportional, when inversely proportional, they cancel out. And that was Galileo's result from the Leaning Tower of Pisa. And that's why The gravitational constant is a constant. They cancel out. So, a successful theory in science. There are lots of successful inferences from universal gravitation. It explains the tides. Its prediction of previously unseen planet, Neptune, the speed of light, a ton of things. It was an enormously successful thing, which is why we study it. Now, I'm sure you know all this, but it is a paradigm for the way reductionist science is done. And I can't think of a better paradigm. So the take-home lessons are the physical world for Newton was like a clock. It's orderly, it's predictable, it's understandable, it's not random. There are no gods exerting their will on things. Of course, the world really isn't a clock like a clock. We know that. But Newtonian model, it is like a clock. And the way the world works, somehow, and this is a really important issue, somehow the way the world seems to work is commensurate with our ability to understand it. Our cognitive ability is commensurate with this phenomena, which is why we can make up laws. And why our cognitive ability is commensurate with phenomena, that's a problem in neuroscience. And it will be explained ultimately. And, you know, we all have our thinking about it. But the fact is, it is. Otherwise, there would have not been Newtonian mechanics. And found model, Newton thought it was weird too. It's counterintuitive, but we can figure it out with Newton's help in the age of reason. So it is important to recognize, and this is part of science too, how the planets move, not why they move. Newton probably meant it when he said, and Newton was a rather arrogant guy apparently, if I've seen further than others, it's by standing on the shoulders of giants. And I'm sure he meant it. So reductionist, just this kind of reductionism, has been the cornerstone of scientific thinking since Galileo. The great scientific revolutions, relativity theory, quantum mechanics, ushered in profound paradigm shifts, but the conviction that an underlying theory, first principles, could account for complex phenomena remained intact. Until now. And now the next one. So I'm going to tell you about AlphaFold. So AlphaFold is an artificial intelligence program, and it is applied to being able to tell you, if you tell me the sequence of a protein, I will tell you the structure of the protein, says AlphaFold. So the protein folding problem, that's the problem I've been working on. You give me the amino acid sequence, and I want to be able to give you the three-dimensional structure. And what we know from lots of work, including in particular work of Anfinsen, is that at least for the simpler proteins, there are complexities, but at least for the simpler proteins, the smaller proteins, that this structure emerges spontaneously from that sequence, without any other factors from the cell. You just take that amino acid sequence. Here are the amino acids in ribonuclease A. There are 124 of them. And if you take a string of those amino acids connected to one another as one long polymer with an N terminal and a C terminal, with a left-hand end and a right-hand end, and you put it in an aqueous environment with a little bit of salt, spontaneously it does that. And so clearly, all the information necessary to encode that three-dimensional structure, which is the basis for the function of the protein, and in particular, the basis for the function of ribonuclease, all of that is encoded in just that amino acid sequence. And the question is, how? We should be able to do it. All the information is there. You put it in water with a little bit of salt, it does that. We know that because its structure has been solved. So that's the problem I've been working on, and you can denature that structure by putting it into some kind of solvent that denatures the solvent. It's not like aqueous environment, and it will unfold. And then when you dialyze away the denaturing solvent, or if you raise the temperature to do this, you lower the temperature to physiological temperature, and spontaneously it does that. So that we're not looking at complicated chemistry. We are looking at a re-equilibration in this equilibrium between the unfolded state and the folded state. Another seeming simplification. It should make this really easy. And yet, using the very best physical chemistry and the very smartest people have been working on this for almost a century. It was first articulated by Linus Pauling and A.E. Mirsky in 1936. So it's almost a century, and it's not been solved by physical chemistry. You would think, but no. So arguably, and I would like to argue it arguably, it's the most important problem at the interface between chemistry and biology. Why? Because just about everything that's done in us is done by some protein. So we really want to know how this works. Now, along comes artificial intelligence. These things like large language models like ChatGPT, which is available to you on your computer, and AlphaFold, also available to you on your computer, and they're making a transformative difference in the way we approach these problems. So I'm going to turn specifically to AlphaFold and the protein folding problem. And so what does AlphaFold do? By combining physical and geometric constraints and patterns from proteins in the protein data bank, these machine learning algorithms can predict protein structure, and they can do it at essentially atomic resolution, which means at really high resolution. You can see individual atoms in the structure. And this accomplishment seems all the more remarkable because very few people saw it coming. It was named Science Magazine's Breakthrough of the Year amongst all science that year. They won. So it's a major advance in protein fold prediction. And what's the PDB? It's 52 years ago. The PDB was started in order to collect the structures and other information about them from proteins as crystallographers solve these proteins. And then as crystallographers and NMR spectroscopists. And then adding to that, eventually, people were doing cryo-EM. But it is a repository of the structures of solved proteins. So it started out with a couple globins and a couple of enzymes. And today, the PDB in its 52nd year has grown to more than 200,000 solved structures. Now, that's really important because protein fold prediction using artificial intelligence has transformed the field by being able to predict the structure from the amino acid sequence. It does it in ways that we have not been successful in doing simply from physical chemistry. And what is it doing? It is using information that comes from the PDB. So we'll come to that in a second. How do we know that it's really successful? There are a couple of ways. I mean, one thing you can do is predict the structure of a protein you know and compare it with that protein and see how well it did. But in order to keep people from cheating, I think, I'm trying to remember how many years. I guess 28 years ago, John Molt and a couple of other people started a contest called CASP. CASP stands for Critical Assessment of Structure Prediction. And in order to keep folks who think that they know how to predict the structure, keep them honest, and it doesn't mean they went out and deliberately cheated, but it's really easy to fool yourself. So they would take crystallographers who were solving proteins or had just solved a protein structure, and they'd ask the crystallographer, please, Mr. Crystallographer, don't tell anybody what this structure is until after we finish this meeting of CASP. And then they take the sequence And they challenged anybody who wanted to enter this to predict the structure. And they would predict it, and they didn't know the structure, but the structure was here behind the curtain. And so in something like 87, there were many entries, people who wanted to do this. And they took the top ones and they used a criterion for assessing how well the structure had been predicted. And this indicates how well the structure was predicted. And AlphaFold also entered, and it blew everybody else away. Shorter line means predicted it. It did it at higher resolution. It did it more successfully. It did it so that the root mean squared difference was smaller between the predicted and the actual. So here it is way back here. It blew everybody away. So that is how we know AlphaFold is really successful. And so these machine learning algorithms can predict protein structures at near atomic resolution. They can do some of them in seconds. And so these computational methods have solved more than 200 million protein structures, all the available proteins from different genomes. And they're accessible. You can download. Here's a database. You want one for your work. You're working on protein X. You want to see what its structure is. You go to the database, download it. So how does it work? In the paper, John Jumper, who was one of the people who was involved in developing AlphaFold, gave this flow chart of how it works. So how does it work? First of all, what's absolutely critical is the structure database, the PDB, with 200,000 solved structures in it. And it goes through some things. It also uses a genetic database, so it gets a lot of sequences. Exactly how this works is, I don't know. I don't understand exactly how it works. But it's clear it goes through, each of these things is a bunch of neural nets. We'll talk about a neural net in a second. But each of them is a bunch of neural nets, and it goes through multiple times, and it has some way of recognizing how likely this structure is. And so it develops a loss function. It's trying to minimize the loss function. And one of the things that is absolutely critical in recognizing how this works is there is absolutely no physical chemistry in it. So it is just based on proteins whose structure you know, and hence patterns that it can extract from that. So there's that. And the other thing is, sounds completely ridiculous. Once it has the sequence, how does it start looking for its structure? It starts the structure by putting all amino acids at a single point and starts to expand them from there. So, I mean, it seems almost ridiculous, but it works. And the reason it works is because, I claim, because it has homologs that it can extract from the genetic database, and it has 200,000 structures from which it can begin to extract patterns. And so there are several neural nets in this flow chart. So what's a neural net? The concept of a neural net is based on our understanding of the human brain. And here it is described in Wolfram's book. Wolfram wrote a very accessible book called What is ChatGPT and Why Does It Matter? And if you're interested in these kinds of things and don't know lots about it, I'm sure there are people in this room who know more about it than I do. But you can get this book. But Wolfram says in human brains, there are about 100 billion neurons, nerve cells, and they're capable of producing electrical pulses up to about 1,000 times a second. And those neurons are connected in a complicated net, with each neuron having tree-like branches allowing to pass electrical signals to perhaps thousands of other neurons. And in a rough approximation, whether any given neuron produces an electrical pulse at any given time slice depends upon what pulses it's received from other neurons, and with different connections contributing different weights. So it's weighted. It's almost like a finite state automaton on in a context-dependent way on exorbitant steroids or something. It gets inputs, it gives outputs. But exactly how this does it is really complicated. And here is the mammalian brain. And there's the, the neocortex. It's the largest part of the brain. It has a folded sheet of neurons. It has 6 layers. They're all interconnected. Excuse me. Artificial neural nets were made to try to model this. That idea of being able to model neural nets started in about the 1940s, and it was never really very successful, although the ideas sound really good. But the reason why it's really successful now is because you have the PDB, you have genetic databases with huge sequences, and you have enormously good computers. So what's a neural net? Here's a toy neural net. It has an input layer, it has an output layer, and it has a hidden layer. And this is a 3-layer neural net. And all of these things are interconnected to one another. And you imagine that these are actually neurons. Only our own neocortex, instead of having 3 layers, has 6 layers. And the AlphaFold has many layers. So it works in an enormously high-dimensional space, AlphaFold, and ChatGPT 2, and it has many billions of parameters, billions and billions of parameters. So here's Holden Thorpe's op-ed piece in Science. It was named Science Magazine's 2021 Breaker of the Year. I said this. But the thing I want to emphasize is that deep learning AI, AlphaFold in particular, is pattern recognition. It isn't physical chemistry. No physical chemistry goes into it whatsoever. It doesn't need physical chemistry in order to do what it does. So let's be very careful in the words we use. It's a major advance in fold prediction, but it's not folding prediction. It doesn't tell us how the protein went from the unfolded state to the folded state. It just tells us what the folded state is if you tell it what the sequence is. Does that matter? Well, we think that we would like to know from first principles how the protein goes from an unfolded state to a folded state. And as I said before, we've been working on this problem for about 90 years, and we still don't understand it from absolute fundamentals, although we have lots and lots of ideas, but they just have not matured to the point that you can do this with physical chemistry. So I belong to a working group that meets every other year in Telluride. It's a coarse graining group. And here are the people who are in this group, and they begin to complain. And they complain because they're all doing coarse grain modeling, but the institute that provides most of the funds for this, the National Institutes of Health, say, well, we're going to really change our focus and start supporting artificial intelligence because it works really well, and you guys don't work that well. And so they're saying, what are we going to do about this? We can't keep working if we don't have support. And so there's a big discussion about it, and all of these people are active, except I'm the only emeritus guy. I'd like to believe I'm still active too, but my lab only has one postdoc, and that's me. So all of these other people have really active labs. So I say, well, why don't we just write something about what we think is successful in AI and what's not so successful, what it's not doing, and why we should still be funded? And they said, that's a great idea, you do it. And I said, well, look, you're the people who are not getting funding that you want. Why don't you do it? And it goes back and forth, but eventually I end up having to do it. And I did it. And so here is an op-ed piece which says protein folds versus protein folding. They're different questions. and there are different challenges. And AI is not doing protein folding, it's doing protein folds. And it can't do protein folding, so I think. Now, if you read current articles about people who are using this, there are lots of arguments that actually learn the underlying physical chemistry, and that's what they're using. And I claim that it's just not the case. And it can't be the case. Why? Because what AlphaFold is doing is extracting patterns from these 200,000 solved structures. But those proteins are already folded. They're fully baked. They're fully pre-organized. Any physical, chemical, organizing interactions were already realized in order to get to that final structure. And all AlphaFold knows is the final structure. So I am really skeptical that it could ever get to the folding problem, but it certainly has mastered the fold problem, although not entirely. So the patterns are extracted from these solved structures. It's essentially a parts list. It circumvents the overall folding process. So had there been no protein data, there would be no AlphaFold. But it's an existence proof that the patterns exist. And if we know that the patterns are there, if we have confidence, that can help us change the way we who do physical chemistry try to approach this problem. So no argument that AlphaFold is incredibly useful. No argument that AlphaFold has done an absolutely stunning thing. But my feeling is we also need, in concert with that, to keep going with basic science approaches. So in perfect analogy to what AlphaFold is doing, well, here's another analogy. It's like watching a movie, and you just watch the last 10 minutes of the movie, and you know how it ends, but you have no idea what the plot is that got you there. But a better analogy maybe is Mendeleev's compilation of the periodic table, which was just based on the properties of the elements. But it was then derived, Pauling derived it from quantum mechanics. So this is an enormous cognitive success. And it is basic science at its highest. So physical science versus machine learning, you try to explain things from first principles and a small number of parameters like universal gravitation, the source of our cognitive ability to describe complex phenomena in this way seems utterly unreasonable, but we do it anyway. Here's a really nice article on the unreasonable effectiveness of mathematics in the natural sciences by Eugen Wigner. And John von Neumann made fun of people who overfit their data using too many parameters. With 4 parameters, I can fit an elephant, he said. With 5, I can make him wiggle his trunk. AlphaFold uses billions of adjustable parameters, so it's a whole different thing. And it's not hypothesis driven. There's no hypothesis. It's just like a newborn child who learns a language just by using the language. The newborn child has no idea what the grammar of the language is, but can speak the language fluently without ever knowing what the grammar is. So I think it's rather like that. So science as we know it, we've been through this, the 3 steps. It's been very successful in the past. Biology hasn't, you'll notice these are all physics things. Biology has lagged behind because unlike physics, it's more complicated. It's self-modifying. It's therefore more complex. In fact, it is much more complex. So here we are. We have solved structures. Deep learning AI has been able to leverage those in order to show us what the structure is from the sequence. Is there going to be a next step here? Is there going to be a theory model step? I think that's an open question at the moment, but I leave you with that question. And thank you so much for inviting me to do this. Now, I'm happy to take questions. I'm incredibly hard of hearing, even with my hearing aids. And what that means is I probably will not be able to understand your question because I won't be able to hear it. But fortunately, I have somebody who has excellent hearing who's volunteered to do this. So it's open for questions. And I can try to understand by approaching it.
Professor Rose, it was wonderful listening to you. Do you think all CASP effort has gone on the basis of 200,000 structures? But what AlphaFold did is having another database, which is millions and millions of genome sequence, and use the evolutionary principles. So biology has evolved with evolution. So how do you make it quantifiable physical chemistry?
I should be a little bit more forthcoming about CASP. So CASP changes its predictions or divides predictions into categories. And so one category is people who are making predictions from sequence, but they're based on what they think are models of similar proteins. And, they're scored separately from somebody who wants to make a prediction based on just first principles. So when I say this, I'm talking about people who've made predictions based on first principles. And you're absolutely right. Evolution has modified these proteins over the course of time. And so this other category where people are trying to make a prediction based on homologs, and they recognize their sequence looks rather like the sequences of some subset of proteins in the genetic database, and some subset of that subset has known structures, then they say, well, I will use these templates to guide my prediction. And that has worked quite successfully, but again, not as successfully as AlphaFold. So, have I answered your question?
Fascinating talks. Absolutely fascinating. And I have several questions, but one question that I would like to ask right now, and maybe we can discuss later outside, other questions, has to do with the fact that you mentioned that Eastern philosophy is slightly different from the Western philosophy. So is it possible for us to understand anything simply by deeply contemplating on the problem, without doing experiments, without going back to any references, past references? Is it possible for us to derive answers for complicated questions like what Newton did and what other people did without ever doing any experiment, or simply by contemplation?
So it's a complicated question. The answer is that I think it's possible to do this without experiments. And that's what we're trying to do with physical chemistry. But, I will claim, yeah, I did it. But how do we know whether I was successful in doing it? The only way is with experiments. So what the Greeks do with thinking is what we do with thinking. It's how Newton came up with Newton's laws. But in order to confirm that that is a meaningful result, you really do need experiments. And so experiment is the true adjudicator of scientific truth. You have to have the experiment. Yes. So the answer to that is yes. And there are a couple of instances in which it's actually been done. So somebody working on, but it's not an easy task. So somebody working on a protein called tryptophan synthase, and he'd been working on it for a long time. You know this protein? Yeah. So he'd been working on it for a long time, and he knew everything biochemically about this protein. And so he started to do something that is basically just like a self-consistency check. I'm going to try to figure out what the structure of this protein is. And I know this is here, and I know this reacts with that, and I know if I put it together this way, I'm going to get a steric clash, and I grew up in the shadow of Ramachandran too, and I can't have that one. And you keep going through this process in your head, and he came up with a structure of tryptophan synthase. And once the structure was solved, he was pretty close. He was not at atomic resolution, but he had a kind of 6 angstrom structure of it. And, Russ Doolittle did the same thing with fibrinogen. Russ knew everything about fibrinogen. And so he began to try to put it together. And he got a pretty good structure for it without ever. He got tired of the fact that crystallographers never solved the structure, and he took crystallography. He went to a graduate course in crystallography with Joe Kraus. He learned how to do crystallography, and he solved the structure himself. So he confirmed his own structure. Yeah.
Yeah. The question is of AI and explainability. Is the distinction between protein fold and protein folding simply the fact that we don't have explainability from these black box neural networks?
Yeah.
And so if that is what is the difference, then the question is, can AI actually get to explainability?
Yeah.
And that would probably need a synthesis of what we now think of as deep learning and logic-based learning, machine learning, which are, till now that synthesis has not occurred.
Yeah.
But it is something that folks in AI are actually thinking about.
Yes, as you say, it is something that people working on AI are thinking about. There are all these hidden layers with billions of connections, and you don't know how it got there, but it is a topic of research. However, even if you were somehow able to penetrate that and know, it still wouldn't tell you how the protein folds. It would tell you how AI managed to recognize the fold. And another thing is that once you see what AI is doing, you can probably, I'm guessing, but you could probably design little experiments with AI in which you didn't try to do some entire protein, but you tried to break it down into some kind of steps and use AI and maybe begin by just thinking about it. You could begin to deduce maybe what it's doing. After all, if what I'm saying is correct, if it's really recognizing patterns, and of course we try to recognize patterns, but the patterns may be more complex than the kind of thing we're looking at. And it may not be really obvious ones. I mean, we look at a pattern, we say, yeah, I see a beta turn, and there's a hydrogen bond here, and I recognize that there tend to be hydrophilic, I recognize that pattern. It may not be something really simple like that. But if we could begin to probe and plumb what the patterns are in AI, then we might be able to make an alphabet of these patterns. And then we might be able to do our own AI. So yeah, it's a good question. And it's the way I think lots of people are approaching this. So.
Taking his question forward, his points, once we have interpretable AI and understand that in science, we should be able to predict. Like we got the biochemical pathway, then we predict the pathway. Then comes the predictive AI. The day predictive AI will design a protein and say this is going to be the function, that day we have understood protein folding. Do you agree with it?
So it might be able to design a protein, and you might be able to design a protein now just by using AI and taking something less than a protein, finding something that seems to work, starting to classify things just by putting the parts together. But it's not telling you why they work. It's telling you that as you've seen many instances in which this verb follows this noun, it's that kind of thing. But it doesn't tell you the rules about how it actually got there. And this issue of interpretable AI, I know people are working on it. You probably, I'm guessing, you know better than I do, but I think we have a long way to go. And will we ever get there? I mean, look, my brain has a neocortex that's a very complicated neural net. At my age, sometimes I forget something that I used to be able to get just like that. I'm trying to remember a name of somebody. And so I can't remember it. Now, often what will happen is 5 minutes later I'll remember it. But sometimes 5 minutes later I don't remember it. And I worry about it and I say, oh, screw it. I'm not going to be able to get this. And you know, you could go to you could go to Google and probably find it. But it becomes a challenge at this stage of the game. I'm not going to do it with Google. And then you go to sleep and you wake up about 2 in the morning and you say, that's it. That's the name. I didn't know I was thinking about that. And it's happened often enough. Now, tell me what those hidden layers were doing. I don't know. So, interpretable AI, I'd like really to have an interpretable real neural net. So, okay, thank you for the question.
Okay. Any other questions? Okay, I have one.
Yeah.
Just last question. So, you know, the folding problem, it seems to me, is not a particularly well posed problem in the sense that depending upon what conditions you have, it can fold through different ways, right? So it's less like the structures can have a few variations, but by and large, it's one structure. But pathways can be any number depending on conditions, pH, temperature, etc.
Yeah.
So it seems to me that, you know, it's going to be very difficult to solve by any method, whether it's by AI or anything else.
Yeah.
Because
so thank you. Yeah, that's a good point. And I've glossed over something when I said this. So when I talk about the protein folding problem, as I did at the beginning, I'm talking about the simplest incarnation of the protein folding problem. Something like, well, the initial one, ribonuclease. And what I'd like to do is to know how ribonuclease folds up into the ribonuclease fold. And it doesn't fold up into other folds. Does it have multiple pathways that it can assume? It doesn't seem to have, it may have multiple pathways in the sense of a couple of pathways, but it's not, I think, it is not like the way that people who represent folding as funnels, and they say, yeah, the folding pathway can go down this funnel any which way, and it just reaches the bottom. So there are tons of different folding pathways. I don't think that that's the way it works. And the reason why is because there's lots of evidence that it doesn't work that way. And when experimentalists, have tried to find out whether there are multiple pathways, they sometimes come up with 2 or 3, but often not even that, and certainly not more than that. And there may be very minor pathways that they can't detect experimentally, granted, but it's typically 1 or 2 or 3. When my daughter was very young, she was precocious, and she could count by the time she was 6 months old, but she couldn't count very high. So she would count 1, 2, 3, whole bunch. And that's the way they're doing this, whole bunch. But we never really know whether whole bunch is there. And so now what you say is absolutely true. There are certainly proteins that fold in a more complicated way. And now we know something that we didn't know then, which is that there are proteins that can adopt multiple different conformations. But the question all along has been, why is a protein a simple one, like ribonuclease, and it starts to fold? Why does it fold to just one conformation, one native conformation, under physiological conditions? Why doesn't it find metastable places along its pathway of folding? Metastable relative to physiological KT. Why don't we see multiple different conformations, intermediates, if you will? But you don't. And you don't even see intermediates. It's either fully folded or fully unfolded for these small, simple proteins. And I think I know the answer to that. At least I have an answer to that. But as I tell people when they raise these kinds of complex questions, I'm interested in trying to solve this problem for simple proteins that I don't understand before I turn to more complex proteins that that I also don't understand. Thank you for the question. So,
Okay, one last question, then we can adjourn for tea. Two last questions.
Thank you. It was nice talk. I just want to ask, is there there will be some techniques to see the folding process? Let's say with the electron microscopy, can we record the snapshots of protein folding, and then we feed into AI to get the remaining parts of the story? So, is it possible, first of all, to get the folding process snapshots with some experimental technique, and those snapshots can be feed into through AI to get the remaining part of the story?
yeah. So, snapshots. Here's the thing about at least these simple proteins. As you start to fold the protein, and you denature the protein, and then you dialyze away the denature it, and the protein starts to fold. And if you're a chemist, you say, as it starts to fold, I'd like to see these snapshots. And then that would be a sort of trail of breadcrumbs, and I could go from the unfolded state to the folded state along this pathway. But they don't work that way. The way they work is they are either fully folded or fully unfolded, with a absolutely negligible population of these intermediates. And you can't detect them. You can only detect them with kinetics. But they just aren't, they're negligibly populated. And why is that? Well, it's telling you that folding is highly cooperative. So it becomes like an all or none process. Physicists looking at it call it a first order phase transition. It really isn't a first order phase transition. Clearly it has to go through intermediates, but they're so negligibly populated that you simply don't see them. So, but with kinetics you can begin to detect them.
Is it a time window we are not able to?
We'll talk. We'll talk. It's a good question.
Last question.
My question is provoked by perhaps an inaccurate parallelism between thoughts. But what it occurred to me that as the protein begins to fold itself, I draw a parallel simile as a ball falling on an inaccurate kind of a topography. And at every point, there is something which is extremized. And that's the Lagrangian or whatever you call it.
At every point, there's something that
extremized.
There's an extreme. There is a function which is extremizing.
Okay. Yeah.
So I was wondering if there is some such function, some thermodynamic function or information theoretic function, which is being extremized as the protein proceeds to form itself.
Well, the answer to that is, personally, I think so. I think what's happening is that you are not seeing something that if you took time slices as a ball falls, it's not like that. It is a highly cooperative thing, but it really does have some extremized points along the way. But what you have to do, I can say it better. So if you're starting to fold the protein and you take some of the polar groups out of the unfolded protein out of water and you put them on what is forming to be the core of the protein, if you had some of those polar groups which could make hydrogen bonds with the water, but they couldn't make hydrogen bonds inside the protein, not just yet. So that would be a stepwise process. You couldn't have that stepwise process. Why? Thermodynamically, why? Because as you took that polar group out of the water and tried to put it in the protein, but it doesn't have a partner, that's at a cost of about 5 kcals per mole. The whole free energy difference between the folded and the unfolded form for typical globular proteins is between about -5 and -15 kcals per mole. And so one unsatisfied bond rivals the whole free energy difference. So how can you begin to do this stepwise step? It really has to fall like the ball all the way to the floor, all at once. And what that means is it's cooperative, and the hydrogen bonds, as it folds, the hydrogen bonds have to be in a folding conformation that allows them to satisfy the hydrogen bond the polar group that you took out of water and put inside the protein. And we know that there are only two ways of doing that for proteins that are deep inside. They can make helices or strands. So there is some changes that are going on there, but those changes I'm sure, or I propose that those changes are ones in which you have to obey the constraints imposed by the necessity of making hydrogen bonds. Yeah.
I realize the inaccuracy of my analogy because this is a constrained situation, unlike the unconstrained situation.
Yeah, yeah. It is a constrained situation, yes. Highly constrained, and that's, I think, what is going on. So, yeah, thank you for the question.
Okay, so thank you. I would like to thank, of course, Professor Rose for an excellent talk, for our chair for coming here and chairing the session, and being with us and telling us about Professor Rose's a lot of his earlier work, and of course the Indian Academy of Sciences for making this happen, and the team over there too, who's ensured that all these arrangements have been made. And now, who gives the shawl? Our chairman.
Thank you so much. Thank you all. This has been a rare privilege.
Okay.